Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's lesson, I'm going to introduce to you the different parts of the cell, particularly the cytoplasm and the cellular organelles of the endomembrane system. So stay tuned. This is a prototypical human cell. While this image is not an indicative of any one particular human cell, it is a prototypical example of a cell containing the primary organelles and internal structures. From my previous video, I've already introduced about the cell membrane, the different function of the cell membrane, the components, and the different activities in the cell membrane. So you can now dive inside the prototypical human cells to learn about internal components and their functions. So all living things in multicellular organism contain an internal cytoplasmic compartment and a nucleus within the cytoplasm. We have this cytosol, the jelly-like substance within the cell that provides the fluid medium necessary for biochemical reactions. Eukaryotic cells, including all animal cells, also contain various cellular organelles. So what is an organelle? An organelle is one of several different types of membrane enclosed bodies in the cell, and each performing a unique function. Just as the various bodily organs work together in harmony to perform all of a human's functions, the many different cellular organelles work together to keep the cell healthy and performing all of its important functions. The organelles and cytosol taken together compose of cell's cytoplasm. So the nucleus is a cell center organelle which contains the cell's DNA. So let's move on to the different organelles of the endomembrane system. A set of three major organelles together form a system within the cell called endomembrane system. And these organelles work together to perform various cellular jobs, including the task of producing, packaging, and exporting certain cellular products. The organelles of the endomembrane system include in endoplasmic reticulum, Golgi Apparatus, and vesicles. So this is a picture of endoplasmic reticulum or ER. Let's take a look at the picture in your left hand portion. That is the figure A. The ER is a winding network of thin membranous sacs found in close association with the cell nucleus, and the smooth and rough endoplasmic reticula are very different in appearance and functions. And in figure B, in the middle of this picture, rough ER is studded with numerous ribosomes, which are sites of protein synthesis. And in the figure C, which is the smooth endoplasmic reticulum, synthesizes phospholipids, steroid hormones, and regulates the concentration of cellular carbon. Also metabolizes some carbohydrates and break down certain toxins. So the endoplasmic reticulum or ER is a system of channels that is continuous with the nuclear membrane, or what we call as the envelope, covering the nucleus and composed of the same lipid bilayer material. The endoplasmic reticulum also provides passages throughout much of the cell that function in transporting, synthesizing, and storing materials. The winding structure of the ER results in a large membranous surface area that supports its many functions. The endoplasmic reticulum can also exist in two forms. One is the rough endoplasmic reticulum, and the second one is the smooth endoplasmic reticulum. So these two types of ER perform some very different functions and can be found in very different amounts depending on the type of cell. So number one, the rough ER is so-called RER because its membrane is dotted with embedded granules or organelles called ribosomes, giving the RER a bumpy appearance. A ribosome is an organelle that serves as the site of protein synthesis. It is also composed of two ribosomal RNA subunits that wrap around the mRNA to start the process of translation, followed by protein synthesis. So, smooth ER or SER lacks these ribosomes. One of the main functions of the smooth endoplasmic reticulum is in the synthesis of lipids. The smooth endoplasmic reticulum synthesizes phospholipids, the main component of biological membranes, as well as steroids, hormones, and for this reason, cells that produce large quantities of such hormones, such as those the female ovaries and male testes, contain large amounts of smooth endoplasmic reticulum. In addition to lipid synthesis, the smooth endoplasmic reticulum also sequesters and regulates the concentration of cellular carbon, a function extremely important in cells of the nervous system where the carbon is the trigger of neurotransmitter release. The smooth endoplasmic reticulum additionally metabolizes some carbohydrates and performs a detoxification role, breaking down certain toxins. In contrast with the smooth endoplasmic reticulum, the primary job of the rough endoplasmic reticulum is the synthesis and modification of protein destined for the cell membrane or for export from the cell. And for this protein synthesis, many ribosomes attach to the ER, giving it the studded appearance of rough endoplasmic reticulum. Typically, a protein is synthesized within the ribosome and released inside the channel of the rough ER, where sugars can be added to it by, of course, a process called glycosylation before it is transported within the vesicle to the next stage in the packaging and shipping process of the Golgi Apparatus. So this is a picture of Golgi Apparatus. In figure 1, the Golgi Apparatus manipulates products from the rough endoplasmic reticulum and also produces new organelles called lysosomes. Proteins and other products of the endoplasmic reticulum are sent to the Golgi Apparatus, which organizes, modifies, packages, and tags them. Some of these products are transported to other areas of the cell and some of them exported from the cell through exocytosis. So enzymatic proteins are packaged as new lysosomes or packaged and sent for fusion with existing lysosomes. And in figure B, it is an electron micrograph of the Golgi Apparatus. So Golgi Apparatus is responsible for sorting, modifying, and shipping off the products that come from the rough endoplasmic reticulum, much like a post office. And the Golgi Apparatus also looks like stacked flattened discs, almost like stacks or oddly shaped pancakes. Like the endoplasmic reticulum, these discs are membranous. The Golgi Apparatus also has two distinct sides, and each with a different role. One side of the apparatus receives products in vesicles, and these products are sorted through the apparatus, and then they are released from the opposite side after being repackaged into new vesicles. And if the product is to be exported from the cell, the vesicle migrates to the cell surface and fuses to the cell membrane, and the cargo is secreted. And the third one is the lysosomes. Some of the protein products packaged by the Golgi include digestive enzymes that are meant to remain inside the cell for use in breaking down certain materials. And the enzyme containing vesicles released by the Golgi may form new lysosomes or fuse with existing lysosomes. So what is a lysosome? A lysosome is an organelle that contains enzymes that break down and digest unneeded cellular components such as damaged organelle. A lysosome also is similar to our wrecking crew that takes down old and unsound buildings in the neighborhood. Autophagy, that means self-eating. That is a process, or autophagy is a process of a cell digesting its own structures. So lysosomes are also important for breaking down foreign materials. For example, when certain immune defense cell, or what we call as the white blood cells, phagocytize bacteria, the bacterial cell is transported into lysosome and digested by the enzymes inside. As one might imagine, such a phagocytic defense cells contain large number of lysosomes. Under certain circumstances, lysosomes also perform a more grand and dire functions. In case of damaged or unhealthy cells, lysosomes can be triggered to open up and release their digestive enzymes into the cytoplasm of the cell, killing the cell. So this self-destruct mechanism is called autolysis and makes the process of cell death controlled, which is a mechanism called apoptosis. So this ends my discussion about the organelles of the endomembrane, particularly with the three major organelles, which are the endoplasmic reticulum, the rough and the soft endoplasmic reticulum, the Golgi Apparatus, and the vesicles and lysosomes. So if you have any question, please write it in the comment section down below and I'll be glad to answer them all. And once again, I would like to thank you all for watching this video and for listening to my discussion. Until my next video, bye everyone!